So thank you, everyone. Thank you to the ISHR and DAVOR really for organizing this fantastic webinar series, especially as an early career researcher. This is a fantastic opportunity, and I know many of us appreciate it. So as Professor Eschenhagen explained, what I'll do for this webinar is take you through some of our recently published data that I worked on as a postdoc with Jeff Mulkinson, where we started with the question of cardiac regeneration and using lineage tracing to assess the role of putative cardiac stem cells, adult cardiac stem cells in this process. And in doing so, we actually ended up finding some pretty surprising and interesting results on the wound healing response, and particularly the role of macrophages in dictating the outcome after ischemic injury. And so I'll take you through these studies. So as we know, the loss of functional myocardium really drives the vast majority of the cardiovascular diseases that we are interested in. And this is in large part because after an injury event in an adult mammalian heart, there's a wave of subsequent myocardial loss, necrosis, and cell death that gives rise to a significant fibrotic response and to a persistent scarring that really is largely thought to be irreversible. And this scarring response and the loss of contractile myocardium is essentially what contributes to ongoing pathology, ultimately culminating in many cases in heart failure. And so what's been known and what's really come about in the last 10 years or so is that in contrast to the adult mammalian heart and to what we see in patients, there are organisms and there are situations where the heart actually has proliferative potential. And in many cases, this is in lower vertebrates, this is in some cases, neonatal animals. And so in this proliferative potential, you actually see not a persistent scar, but subsequently, after injury, you see inflammation, angiogenesis, and proliferation of myocytes, and ultimately, in the most extreme examples, complete restoration of function. So why does the adult mammalian heart not regenerate? And so this is obviously a very large question, but really the key concept is that the cardiac myocyte in the adult mammalian heart is minimally, has minimal renewal capacity. And so what's shown here are some of the key data in humans in post-mortem samples taken from basically retrospective carbon-14 dating studies, which have shown that in human hearts, the turnover estimated rate of cardiac myocytes is about 1% per year. And this is actually a measure of DNA synthesis, and so true myocyte division might be even lower than this. And in the same case, in model systems, including in mice, we and others have shown using genetic lineage tracing, which I'll talk about in a second, that rodent hearts have a pretty low regenerative capacity and turnover of cardiac myocytes as well. And so taking all this into account, if we really think about this in an overly simplistic perspective, this is essentially a numbers problem, in that compared to a healthy heart, the injured or failing heart has a deficiency of functional contractile cardiac myocytes. And as we know, many, if not all, of the current therapies available to us as a field, including LVAD, mechanical support, and various pharmacological supports, really do not address this fundamental loss of functional cardiomyocytes, and instead, really anything short of heart transplant cannot address this to any meaningful extent. And so the field really, for a long time, has searched for therapeutic approaches which can directly address the loss of functional cardiomyocytes in a meaningful way. And there are a lot of ways that this has been explored, and there's some really exciting results coming out of multiple labs which have looked for ways to actually induce existing cardiomyocytes to re-enter the cell cycle, to transdifferentiate or transplant iPS-derived myocytes either as single cells or as tissue scaffolds or engineered heart tissues. And there's also technologies that are being explored to reprogram non-myocytes into myocytes using direct reprogramming of fibroblasts, or other exploratory options, including tissue hypoxia and other extracellular cues. But what really took hold of the field about 2 decades ago now was this other concept that the heart can be regenerated from adult tissue stem cells. And so for the purpose of this talk, when I talk about a stem cell, I'm talking about this sort of collection of adult tissue resident cells that have been proposed to have stem or progenitor-like properties. And many of these are shown here on the right. Many of them come from the bone marrow or from the bone marrow stroma as mesenchymal cells. There are a number of cells that have been proposed to have stem-like properties coming from the heart itself, and I'll talk a little bit about these various sources. And the take-home message is that all of these cell types at one time or other were proposed to be able to regenerate substantial amounts of myocardium, but more broadly have been shown to have functional benefit when delivered into an injured heart. And so those are the key questions that we became interested in a few years ago. In part because this has been a highly contentious and controversial topic in the field. And so for many, many years, there's been this ongoing debate about the rationale behind cell therapy, behind why so many clinical trials of this have failed, and why in the preclinical space there seems to be data in animals that this might be a meaningful way to at least repair heart function. So this is really where we jumped in a few years ago now. And what I'll talk about are a couple studies that really try to address these major questions. So what is the contribution of these adult cardiac resident stem cells to endogenous myocyte renewal or turnover? And I'll talk about how we've used genetic lineage tracing to address this in a mouse model. The second sort of broader question, how is it that so many different types of cells that have progenitor-like function or have been claimed to have progenitor-like function, how can they all contribute to this functional improvement in animal models that has been seen? And then sort of giving away the ending, in the absence of true regeneration, what are the pathways that could be impinged upon to govern and improve cardiac tissue healing? And that's where we think the immune response and specifically the macrophage response is really an interesting way to go. So before I get into the data, I'll briefly introduce this concept of genetic lineage tracing, because these data will be a major part of what I show you. And so in a nutshell, this is a transgenic mouse system where you have a driver allele that can actually be expressed in a cell type of interest. And usually this is a form of Cre recombinase, either constitutive or inducible. And then you also have in these animals a genetic reporter allele, which is usually inserted into a ubiquitously expressed locus, which is responsive to Cre recombination. And then when this cell type is actually induced to recombine, what happens is there's a genetic fluorescence reporter that's expressed in that cell, and that reporter is persistent throughout the lifetime of that cell. So what that means is that you can label a population of cells over time, and then their progeny, even if they no longer express the driver, will stay labeled. So you can track their long-term identity in vivo. And so what you see a lot of times with these sort of data are immunofluorescence images like this. And so this is a GFP positive myocyte that was labeled with one of our genetic lineage tracing systems. And so I'll show you some data where we've used this to actually ask the question of whether some of these populations of cells expressing stem-like markers are actually myocyte-producing stem cells in vivo. And so when I first joined the Wilkinson lab, we began using this to look at c-Kit cells. And c-Kit cells, as Professor Eschenhagen mentioned, were one of the leading candidates for this concept of an adult cardiac stem cell. And so we generated a system where we induced a MER transgene through the endogenous expression of c-Kit and crossed this to mice expressing this Cre-responsive GFP. So essentially, when a cell expresses c-Kit and when tamoxifen is on board to activate the system, that cell turns green and stays green even as it divides and gives rise to new cell types. And so the take-home message from this study is that essentially we saw a very low level of myocytes derived from c-Kit cells, about a fraction of a percent throughout postnatal life and after injury as well. So we basically see from this that c-Kit cells are not a significant source of myocyte production. And so this opened the first series of questions that I worked on, which was, are there other types of cells that have been proposed to be myogenic stem cells? And is this indeed how these cells function in vivo in the mouse heart? And so we focused on a population of cells expressing this stem cell antigen 1 or SCA1. And this is a GPI membrane anchored protein. Much like c-Kit, it came about in the field largely because it's expressed in hematopoietic stem cells and early committed progenitors. And so the concept was, since we know the bone marrow is a robust source of stem cells and these markers are expressed there, perhaps there are similar cell types in the heart. And so this really became a concept now about 2 decades ago. And so there were a number of papers suggesting that SCA1 expressing cells could give rise to cardiac myocytes in various systems and various populations. But we wanted to really use genetic lineage tracing to address this because it gives us a unique ability to mark the intrinsic population of cells without overt manipulation, without transplant. And so we asked the question, what is the functional contribution of these SCA1 expressing cells during cardiac homeostasis or after injury? And so to ask these questions, we generated 2 transgenic mouse lines. Again, we used Cre recombinase inserted into the endogenous SCA1 promoter region. This is LY6A is the gene. And we crossed these mice to a Cre responsive reporter, in this case expressing TD tomato. And so in this first series of experiments, we can constitutively label SCA1 cells and their progeny all throughout development, postnatal growth, or after injury. Then we generated a second system where instead of Cre, we have a tamoxifen responsive mercury-mer. So this gives us temporal control, where we can actually label these cells discretely after birth or at different time points. And here we just use a GFP green fluorescence reporter instead. And in some data I don't have time to show you, we validated these systems using bone marrow and other known sources of SCA1 expression to confirm that they were indeed labeling the cell types, the appropriate cell types of interest. So first, looking at the constitutive system. So here we have mice where essentially Cre is active all throughout birth and postnatal growth. And what we saw with these animals essentially is that starting around birth, around P1, and continuing out throughout the first 3 months of life, we saw an expansion of TD tomato labeled cells, which are SCA1 derived cells. And as you can see, by 3 months of age, there's numerous TD tomato labeled cells throughout the myocardium, and this is simply at baseline. What I think you might be able to appreciate just from the morphology is that these are cardiac endothelial cells. And indeed, when we looked at this with immunofluorescence and with immunostaining, we saw that the vast majority of SCA1 labeled cells in the mouse heart during postnatal growth were cardiac endothelial cells. And this was the case at birth and also at 1.5 months of age. So what about the myocytes? So in this case, we looked at 3 months of age. Again, Cre is active all throughout this time course. So anytime a SCA1 cell is labeled at any point throughout this time course, we'll be able to track it. And again, using co-labeling of both alpha-actinin and PCM1 to mark the myocyte nuclei, we were able to detect a very low percentage of TD tomato positive myocytes, so 0.003% by 3 months of age. I should point out that this does not account for cell fusion events, which are known to occur, for example, with bone marrow cells. And so one possibility is that a non-myocyte expressing SCA1 would fuse with an existing myocyte. And so likely this fraction is even lower. Similarly with the inducible system here, we can actually selectively label SCA1 cells only after birth. And so we can we can distinguish the developmental from the postnatal window of SCA1 activity. And so what we did here is we used our mercury-mer tamoxifen inducible system crossed with a GFP reporter. We induced tamoxifens in chow starting at 3 months of age and left them on chow essentially out to a year. And so we're capturing this entire postnatal window without looking developmentally. And essentially we see the same thing. We see a robust labeling of endothelial cells with GFP and a very low percent, 0.001% GFP myocytes, suggesting that SCA1 cells are not a physiologically meaningful source of new myocytes in the heart. Interestingly, we also did an MI experiment. So we did permanent coronary artery ligation, followed with a pulse of tamoxifen for 4 weeks. And essentially, we tracked those cells out post-MI after tamoxifen was withdrawn. And so what we did find is that we did not see an increase in myocyte labeling. But what we actually saw that was somewhat interesting is that the SCA1 labeled endothelial cells, and with this pulse, we labeled about 20%, and this is quantified over here. This percentage of SCA1 labeled endothelial cells actually expanded post-MI, even though the tamoxifen was withdrawn. So suggesting perhaps that this population of SCA1 labeled endothelial cells might be either more proliferative or might have other properties that would preferentially allow them to expand after injury. To summarize this first part, we found that genetically labeling SCA1 cells showed us that these cells do not give rise to cardiac myocytes, either at baseline or after injury in mice. They do seem to label a proportion of endothelial cells which might have unique properties, which is something we're following up on. And as mentioned, this was part of a series of 5 papers where multiple labs used this technique and essentially showed the same effect. So switching gears, if the heart doesn't contain a population of myogenic stem cells, and I'm not showing data from a number of other groups that had come out after this, which used other methods, including labeling proliferative cells or labeling all non-myocytes, collectively showing that not only just c-KIT and SCA1, but really there is no evidence for a myocyte producing an adult tissue stem cell. And so if that's the case, how are all these different types of cells showing a functional improvement in an injured heart, at least in animal models? And so, what I want you to take away from this is just the diversity of all these different cell types. And so there's things derived from the bone marrow, as I mentioned, from the heart itself, from circulation, and they all seem to have a pretty similar profile, and that they were originally claimed to make cardiomyocytes. This didn't pan out, but they did show a functional improvement after injury. And many labs have shown this, and I'll show you that we see it as well. So how do we explain this? What's the potential mechanism to explain how so many of these different cell types can all have a meaningful therapeutic effect? And so if we can understand this, we might be able to actually optimize cell therapy in ways that would allow it to be more useful. So we set out to address this question, and essentially what we did is we started with a pretty simple experiment. We just took these various types of cells, and I'm going to show you data mostly from bone marrow mononuclear cells. So whole bone marrow MNCs. We focused on these in part because these are the cells that have been used in a large number of clinical trials and were sort of the prototype cell for this concept. So we took these various cell types and we isolated them from genetic constitutive rosa tomato mice. And so in this case, these cells are always labeled red. So we can track their fate over time, similar to what I showed you previously, but in this case, it's a permanent label. And so we injected these into healthy wild type mice at defined sites along the left ventricle, along the anterior wall. And we injected these and we just looked over time to what happens to the cells and what function, what physiological effects do they have. And again, I don't have time to show you all the data. We looked at things like myocyte proliferation. We looked for new vessel formation. We really didn't see anything there. But what we saw, which was really the most pronounced effect, is that wherever we saw these cells injected into the heart, we saw a robust induction of inflammation. And we saw this really pronounced accumulation of activated macrophages, which are marked here with this CD68 antibody in green. And so we quantified this over time, looking out to 2 weeks. And what you can see again is what's highlighted here are these injection areas. And so all of our controls for these experiments are sterile saline. And so saline itself, when injected, does give you a little inflammatory response. It's essentially a needle wound. And so all of our experiments are compared to this state. So here's the MNCs. You can see a robust induction of CD68 macrophages around the area where the cells are injected. And this really persisted for about 3 to 7 days, which is quantified here on the right. And by 2 weeks, we saw that the inflammatory response was largely attenuated, and this correlated with the clearance of the cells. So by 2 weeks, we really couldn't find these cells anymore. This is at 3 days, and you can see where these are. So if the major effect of injecting these cell types is a localized immune response, we wanted to ask the question, is that an important component to their potential effect? And so to do this, we needed a model where we could mimic the inflammatory effects of cell therapy and in a distinct way from actually using the cells themselves. And so what we settled on was a molecule called zymosin, and this is a toll-like receptor agonist. Essentially, it's derived from yeast, and it mimics a sterile immune response. And so we've used this molecule, and again, we were able to get zymosin that was conjugated red, so we could track those particles. And we decided to test whether this simply inducing inflammation would give us a similar profile. And so we did the same series of experiments in healthy mice where we injected Zymacin into the LV. We looked at them out over time, and you can see, much like MNCs, the areas where we injected Zymacin acutely into an otherwise healthy heart gave us a robust induction of CD68 macrophages. And the quantitation showed that the profile was similar to MNCs, and that within the first 3 to 7 days, you get an upregulation of CD68 cells in the injection area, and this is largely cleared by 2 weeks. So now we have a model where we can actually mimic the inflammatory effects of cell therapy without the cells. And so this is really the key experiment, what happens after injury. And so here we took, again, wild-type mice. We gave them ischemia reperfusion, a 2-hour ischemic window with temporary coronary ligation and then reperfusion. And then we decided to wait a week and then open up the animals again and then inject them in the infarct border zone with either MNCs, Zymacin, or sterile saline. And we did this in part to sort of better mimic what would be a clinically relevant scenario of an acute MI patient potentially getting a cell therapy. And so we did our injections around the infarct 1 week after IR. We waited another 2 or 8 weeks and assessed the mice by echocardiography. So this is really the key piece of data. If you look at fractional shortening by echo compared to sham-treated control animals, IR gives a significant decline in cardiac function, as you would expect. And this was significantly attenuated with MNCs. So similar to what many other groups have shown, injecting these cells does give a functional benefit. But we see the same benefit if we inject Zymacin, this inflammatory compound that it has no cellular material, essentially. And that was at 2 weeks post-therapy, and the same effect is seen by 8 weeks. So this is a persistent benefit with either injecting MNCs or Zymacin. And I think if you compare these two graphs, you can see that the functional recovery does decline. So this is not a permanent benefit. This is not something that, it continues to keep the heart functioning normally. They're still declining, but they're declining at a slower rate than the saline group. So we decided to next ask, well, is that inflammatory response that we saw, is that a key mechanistic contributor to this benefit? And so we did a couple of experiments, and essentially the paradigm I just showed you is the same. Again, wild-type mice, IR injury, and then injected 1 week later with MNCs or Zymacin. But we did a couple modifications to test whether inflammation was key to this effect. The first thing we did is we simply injected freeze-killed cells. And so what we've decided to test was if these cells needed to be viable upon delivery, and as you can see, they don't. So if we took MNCs and we simply freeze-thawed them multiple times to lyse the cells, and then injected that dead cell slurry, it was equally as protective as the living cells. The next thing we did is we used broad-spectrum immunosuppression with high-dose cyclosporin. And so we did this, and this is important, we actually started cyclosporin after injury, right before injection, so as not to impair the infarct response. And we used a dose that was high enough to impact both acute, innate, and adaptive immunity. So this is a heavy hammer immunosuppression. And with cyclosporin on board, MNCs and Zymacin failed to give a protection compared to saline. So it's again suggesting that this immune response is absolutely critical. Cyclosporin obviously is going to affect a lot of different cell types, T cells, macrophages, et cetera. And given what we saw with our initial experiments where we saw that induction of macrophages, we wanted to know if they were the key players. And so what we did is we used clodronate liposomes, which is an encapsulated toxin, which can be used to acutely deplete macrophages in vivo. So essentially, we did our same experiment with IR, and then right before our injection, we depleted macrophages with clodronate liposomes. And what you can see, again, here, there's the controls. IR injury still gives us a significant decline in function. The MNCs are still effective, but with liposomes depleting the macrophages, MNCs no longer improve function. To collectively suggesting that this macrophage induction is the key mechanistic component of how cell therapy is acting to improve function. And this is part of a larger concept that's been well described in the field, in that during an acute injury or during an infarct, for example, the heart has a multiphasic acute immune response that's been well characterized, which is schematized here. And you can see an immediate influx of neutrophils usually occurs in the mouse around day 1 after injury. And then there's this biphasic and really intricate macrophage response, which occurs during the next 3 to 7 days. And it's characterized by an early accumulation of inflammatory monocytes, which are largely acting through the CCR2 chemokine receptor. And then there's this second wave of what are thought to be reparative macrophages, or even tissue remodeling macrophages. And this is really where I'm becoming more and more interested. But these cells are largely characterized by expression of another chemokine receptor called CX3CR1. And so we wanted to know if this paradigm was also reoccurring when we used cell therapy to induce inflammation. And so to ask this question, we generated compound heterozygous mice, which have a knock-in of GFP at the CX3CR1 locus, versus, and as well as RFP at the CCR2 locus. So essentially what this lets us do is it lets us label and track these various populations of macrophages in real time. So these are not lineage tracing markers, these are real-time expression markers. And what we saw in these hearts at baseline without any injury is that they predominantly have CX3CR1 green macrophages, as you would expect. So these are thought to be largely the tissue-resident macrophage pool that's derived from embryogenesis. And they really are largely devoid of CCR2 expressing inflammatory monocytes and macrophages. And so when we took these animals and we injected MNCs, and this is into a healthy heart, we saw that this wound healing cascade is entirely recapitulated, in that initially when you inject MNCs, you see this accumulation of CCR2 positive inflammatory monocytes and macrophages within the first day. And then by 3 days, you start to see this mixed population of both CCR2 and CX3CR1 expressing cells. And this is sort of that switching point that's thought to occur as the wound resolves. So we wanted to know if this was a functionally important contributor to our cell therapy effect. And so we next went on and we made global knockout crosses to generate functional nulls for either CCR2 for these inflammatory monocytes, or for the CX3CR1 receptor, which is thought to be a key component of reparative tissue macrophages. And we did the same experiment with these knockouts, where we used IR injury, and then 1 week later injected them with MNCs. And again, the sort of key piece of data, if you look at the wild type, it's the same effect. MNCs restore function in the wild type compared to saline. But CCR2 mice actually already show a protection irrespective of cell therapy, and this has been published before, so this is not surprising. But the important thing is that cell therapy in the CCR2 knockout background didn't really add anything. It didn't improve this functional benefit any further. In contrast, the CX3CR1 knockout mice, they failed to respond to cell therapy at all. And so without these reparative macrophages, MNC cell therapy, much like we saw with acute immunosuppression, failed to improve function. So again, suggesting that these reparative tissue macrophages are a key component to the response of the heart to cell therapy. So how would inducing inflammation and causing this sort of accumulation of macrophages improve heart function? And so we looked at a number of things, and what we think, and we're still working on this, but what we think is a key component is the fibrotic response. And so when we looked at these MNC-treated hearts after injury, we actually saw a significant reduction in border zone fibrosis, as shown here by P-Series red staining. And this was accompanied by a global reduction in gene expression in these infarcts, these cell therapy-treated infarcts, of major ECM and ECM-associated matrix genes, including type 1 and 3 collagen, fibronectin, and periostin. And then to go a step further, we did a collaboration with Sakti Sadiapin's lab, where we actually tested the biomechanical properties of these infarcts. And so to do this, we actually dissected the infarct strip from the mouse, and we put it on a muscle force transducing rig. And essentially what we did is a passive length stretch protocol, where we tested the ability of these tissue strips to induce passive force over increasing length. And so the blue here are the cell therapy treated infarcts versus the red. And just shown here is the data from an uninjured wild type heart. And what we can appreciate is that cell therapy restored the kinetic profile of these infarct strips, so that they were more similar to wild type or to uninjured. Again, suggesting that this change in ECM might actually be imparting functional alterations that might contribute to the improvement in function that we're seeing. And so how would this occur? When we think that this is in part due to direct effects of the macrophage on the cardiac fibroblast. And so what we did is we actually took IR injured mice and we isolated either those CCR2 inflammatory macrophages or CX3 positive macrophages. And then we co-cultured these with primary cardiac fibroblasts. And we looked for changes in fibroblast gene expression. And what we found, interestingly, is that these macrophage subtypes actually imparted quite distinct effects on the cardiac fibroblast, in that the CCR2 inflammatory macrophages induced higher levels of profibrotic genes, including lysyl oxidase, smooth muscle actin, and to some extent collagens. Whereas the CX3-CR1 cells, not only did they not induce fibrosis or fibroblast expressing genes, but they may have mildly suppressed it. Again, suggesting that these macrophage subtypes can quite distinctly change the biology of the cardiac fibroblast. And this is something that I'm actively pursuing in the future. And so the last little bit of data I'll share with you. When we started this, we really wanted to know not only how the mechanism of cell therapy might be working, but how to explain some of the discrepancy between the preclinical data and the clinical trials that have been done with these various cell types. And there's a lot of reasons that might be at play here, but one of the key ones we think is actually the delivery method. And so our data in these rodent models suggests that the immune response is really important, given that you are injecting cells directly into the muscle. In contrast, a lot of the clinical trials, so this is starting to shift a little bit, but many clinical trials have been done with systemic infusion, where these cells were either injected via coronary circulation or other systemic methods. And the idea was that they would perhaps home to the heart and actually improve function that way. And so we wanted to test that concept. So we used, again, our rosa tomato labeled MNCs. We used wild type mice with IR injury, and then we infused 1 million of these cells via the tail vein. And the first thing we asked is, can we find them? Do they make it to the heart? Are they retained in the tissue or in circulation? And so since these are fluorescently labeled, we can track them with flow cytometry and microscopy. What we found essentially is that as early as 5 minutes post-infusion, we were only able to detect less than 0.1% M-tomato positive cells in circulation. And by 2 days, this really didn't change. In addition, if you looked at the heart, the lung, and many other tissues, we were only able to find about 1 to 2 cells per milligram in all of these tissues. So the vast majority of these million cells were cleared in minutes. When we did find MNCs in the heart, and they were very rare, they were always encapsulated within the coronary circulation. So they never really made it into the heart parenchyma. They were always just circulating in the vessels. And this is something we saw not only in heart, but in lung, liver, kidney, and spleen as early as 5 minutes post-infusion. So then the final piece of data is to compare cell infusion versus injection. And so what we did is we looked again at our mice that had received 1 million cells IV versus mice that received our direct injection into the border zone. And we did this at 1/10 the dose, so 1 times 10 to the 5th cells. And what you can see is that, much like I showed you earlier, direct injection did give us a modest but significant improvement in cardiac function, and even a little bit of an attenuation of cardiac hypertrophy. But we didn't see this with infusion, even though we were using 10 times as many cells. So to wrap things up, we think that despite no new myocytes, you can get a functional benefit with these various cell types, but it seems to be the way this works is that all these cells actually induce a common immune response through the induction of macrophages and through the production of these damage-associated molecular patterns, or DAMPs, as the cells die. And we think that this actually explains why so many different cell types, or not using a cell type at all, using just an induction of inflammation, can at least in part restore cardiac function in an injured rodent heart. And we think mechanistically this has something to do with how this immune response reconditions the ECM, the extracellular matrix, and can change things like scar elasticity in part by regulating the cardiac fibroblast. And our data does not preclude that paracrine factors and other mechanisms might be at play. But I will point out that a localized immune response without cells in our hands was sufficient to induce this response. And so overall, we think that cardiac cell therapy works by reactivating the heart's own intrinsic wound healing cascade. And this is something that we think actually could be harnessed therapeutically. But beyond that, these data really open up an interesting hypothesis that's been put forth in the field, and that is that we know that there are components of the immune response that are beneficial. We know that you need a certain amount of inflammation to induce healing. But what our data suggests is that you can actually augment this, that you can induce a little bit of inflammation to cause a net functional benefit. And so this is what I'm interested in pursuing long term. And obviously, it's a very complex question. It has to do not only with the duration, but also the cell types involved. I didn't talk at all about other immune cell types, including neutrophils and T cells, dendritic cells, all can play a role here. So this is incredibly complex, but also important because it suggests potentially that you can actually modulate inflammation to improve wound healing and perhaps, if not regenerate the heart, maybe rejuvenate it or repair it. So that's my conclusion. I'll thank Jeff for all of his many years of support, all the members of the Mokenton Lab. The folks in white were the ones who were directly involved with the studies I showed you. Also our collaborators in Cincinnati, at Temple, at Harvard, and funding sources for this project and others. And I would thank you all again. I really appreciate the platform. Happy to read some questions.
Thank you very much, Ron, and congratulations. We can't clap here, but it was a great talk I found, and it's interesting. I think our discussion will go around this point a bit. Some of the things you found have been speculated about or described previously, but what I really appreciate in your work is a consequence in which you nailed it down and really put the point on, really put the story firm. I think that was really super important and good. So,
thank you.
Do you want to go through the many questions?
Sure.
Try giving answers.
I will start from the top. So I'll read out the questions. Angelo Calderon asks, did you confirm that the DT tomato staining was also SCA1 positive? So that's a good point, and I didn't get the chance to talk about it in detail, but this is something that has been shown in the literature, but is, I think, underappreciated, and that is that about 3/4 of the existing cardiac endothelium expresses SCA1 constitutively. So many of the endothelial cells that we were looking at were SCA1 expressing endothelial cells. And so the question really is what's the functional relevance of that? And there have been some groups that have looked at SCA1 knockout mice. They don't seem to have an overt endothelial phenotype. So my interpretation is that the functional effects of SCA1 are unclear, but we do think that a lot of those labeled endothelial cells were pre-existing endothelial cells that were expressing the protein, as opposed to an expansion, a vasculogenic expansion. So similar question, Bukita Loya asks, with cardiac endothelial cells, do you mean endocardial cells? No, we're talking about vascular endothelial cells, not endocardial. We didn't look at endocardial markers specifically. How widely, anonymous person asks, how widely standardized and accepted across the cardio research community is the use of glycogen synthase kinase beta antagonist followed by Wnt pathway inhibition with chemical IWP2? I think this is referring to IPS differentiation of myocytes. I can't speak on how standardized and accepted it is. I'm not an IPS person, but I have seen flavors of this protocol for IPS.
Oh yeah, I mean, it is actually very widely used, but not by many. Overall, I think this differentiation protocol are either based on the small molecule antagonist of GSK beta or on growth factors. We, for example, use growth factors, not this inhibitor.
Pearl Quijada, hi Pearl. Does your SCA1 lineage tracing label CSPG4 positive or PDGFR beta positive cardiac parasites? You know, we didn't look at parasites. We looked at almost every other marker. And so we do label a population of PDGFR alpha cells. We haven't looked at parasite markers. It's a good question. Something we can look at. Let's see. Deepa Rani, what is the success rate of cardiac stem cell therapy in clinical trials? Well, that depends on the cell type, whether it's phase 1, 2, or 3. I think that's the big question, and I would argue that there are some signs of efficacy, but the net, if you look across everything, it's not, it seems like it's not really working as well as it could. But that's, I mean, there's a lot to be said about that. Angela also asked, did immunosuppression lead to scar thinning? Actually, not in the way we did it. And that's something I was a little surprised at, given what we know about cyclosporin effects and other immunosuppressant effects. But I will point out, we didn't start cyclosporin until essentially a week post-IR. And so at least what we think is that by limiting cyclosporin only to that window of time after scar formation, we may have avoided some of that. But at least from what we saw in those, in those initial 2 weeks, is we didn't seem to cause further scar rupture or scar thinning. Good question. Emilio Medei asked, did you try injecting cytokines like IL-1 beta? Not yet. That's actually something that we're thinking about in terms of what are the key molecules that are important here. And so we know that things like IL-1 beta are often upregulated in heart failure and other types of inflammatory injury. I think it's going to be more complex than simply injecting a cytokine, and I think that's why modulating the macrophage or other immune cell types might be a more pleiotropic way to do this. But we haven't tried injecting other stimuli yet. So like Chen asks, most of the cell therapies were performed in nude mice or rats, and they showed therapeutic efficacy. Does this immunodeficiency affect the inflammation-induced cardiac repair? So there are absolutely studies that have been done with nude mice or rats, and this is something that we talk about a lot. Those animals are not globally they don't have an absent macrophage response. If they did, they wouldn't survive an infarct. So immunosuppression with nude mice, actually, it suppresses macrophage activity. It doesn't eliminate it. And so we still think that this mechanism could still be at play to some extent. Even with the most heavily genetically immunocompromised animals, triple rag, skid, knockout, null mice, they still have to have some amount of macrophage activity, because we know if you completely remove that, the animals would rupture immediately. And we've tested this with some pharmacological compounds.
Can I step in here? Because I think that touches upon a really important question. I'm not so sure whether the majority of bone marrow cell therapy trials, adipose, SCAR1, whatever, have been done in nude mice or rats. I think many, at least the initial ones have been done in splenectomized mice, but not necessarily immunodeficient. And I think most people who use nude mice or rats use pluripotent stem cell derived myocyte. That's another issue. Would you agree that, I mean, some people using IPS or embryonic stem cell derived myocytes injection or patches, all of these things are done either in immunodeficient mice or rats, or under the use of immunosuppressant like cyclosporin A. And would you think that this is an evidence for having a real regeneration in these models?
Yeah, absolutely. I think that, the whole concept around using nude mice with adult stem cells was based on the notion that they would engraft and that they would persist. And we don't see that. In cases where you actually do have engraftment, that you do have perhaps contractile and direct contractile contribution from a tissue graft, then immunosuppression becomes, it's a double-edged sword. So there it becomes a problem to have too much inflammation, because that's how the graft might be lost. So yeah, I think the concept was based around the notion that the adult stem cells would persist, and it seems like they don't, and they may not even persist long enough to mount more of an adaptive immune response. And so I think that's absolutely the case.
Ron, can I just ask quickly, just
Sure.
to kind of interject quickly? I mean, I've seen that you worked with this on this with Matthias Narendorf. Have you detected any changes in conduction or any blocks, or seen any arrhythmias in these mice?
No, but I would say we certainly have not looked extensively.
Okay.
I know the work you're talking about. It's really, really fascinating. We haven't looked at that. We're just starting to dig into the tissue macrophage and what its physiological functions are. It seems like one function might be to conduct current, but yeah, we haven't looked at that here.
Thank you.
Let's see, which gap junction do you consider responsible for the interaction between fibroblasts and cardiomyocytes? I haven't looked at this personally myself. I've seen some interesting data about some work from Peter Cole and some others that have looked at connections. It connects in hemichannels. It's not something we've really looked at personally. I think I've seen connexin 40 might be involved, but again, haven't looked at that much myself. V. Pavrani asks why only MMP3 was examined, why not MMP2 and MMP9 or casinases, which are well reported in cardiac context. Yeah, good. So we're in the process of looking more extensively at the extracellular matrix, and so we picked some candidate genes to sort of profile, but what we're really trying to do now is a more global study where we really assess the matrix, not just by gene expression, but also by protein content. So we're working on that. Good question. Meow Kui says, do you see a reduction of cardiomyocyte death over time with cell therapy? In our model, we don't really see that, and we think some of that might be the way we did the experiments. And so there have certainly been studies where cells or other materials were injected immediately upon coronary ligation. And so that's more of a cardioprotective protocol. What we've done is we've waited a week, and so the majority of infarct formation occurs in that week. That's not to say there might not be secondary myocyte loss, but we didn't see anything to suggest that our therapies were affecting that. Emma Robinson says, I often wonder how much we know about either the endogenous or exogenous induced stem cells for regenerative therapy with regards to their druggable receptor expression. Eventually, patients that would receive stem cell therapy will be on beta blockers, ACE inhibitors, ARBs, etc. How might these stem cells also respond in the presence of these core medications for heart failure? Yeah, great question. That's something that, and I have looked around for this and I haven't seen a lot of data on this, but what I'm interested in addition to those points, is the immune status. And so, it's tempting to wonder if you have a patient with compromised or either compromised or exaggerated inflammation, as we know occurs in heart failure, occurs throughout the sort of progression of ischemia. That's going to change how that patient responds to pretty much any kind of intervention. And so one of the likely possibilities of why there's such a discrepancy between preclinical, early clinical, late clinical is probably that exact point. That you're dealing with highly heterogeneous patients, and it's also been argued highly heterogeneous cells if it's an autologous transplant. So yeah, I think that's definitely a possibility. Something we would have to take some time to figure out. Anonymous attendee: the protective effect of cell replacement therapy is lost in CX3CR1 knockout mice. Is this due to a loss of function, or is there an expansion or persistence of CCR2 positive macrophages? So this is, and I was debating showing some of this. This is actually what I'm working on right now. What we think, and I can only speculate, is we think that the CX3CR1 deletion in macrophages impairs their function. That's what I'm working on. We have a lot of hopefully we'll have some cool data for this to share. We did not see an expansion of CCR2 positive macrophages, but has been shown that if you deplete CX3CR1 in other contexts, including angiotensin-induced fibrosis, you do see that. So it could be both, but we're working on the hypothesis that the CX3CR1 deletion actually does impair the function of the macrophage. It does not impair their numbers, and so we have data showing that when you deplete CX3CR1, you still see those cells in the myocardium. So it's not really a homing factor that changes the recruitment of CX3CR1 cells. They're still there to equal levels. So we think it's actually a functional problem. Richard Heads: if you inject CX3CR1 cells, they have positive effect as opposed to CCR2 pro-inflammatory cells. But what about TRMs? I think tissue-resident macrophages, I'm assuming. How do you see those? It's a small population, but they're already there and they have a very beneficial role. My lab is really interested in this. Yeah, that's a great question. And one of the the things that we're interested in is this idea that when you have an infarct, you do lose tissue-resident macrophages because they will die along with the myocytes and the other cell types. And what seems to be the case is that those cells are replaced by monocytes and replaced by circulating macrophage sources, which there's some really cool data to show that those cells might actually be not only more inflammatory, but actually less efficient at repair. And so what happens after an infarct is you actually shift the whole profile, not just in terms of what the markers are, but actually the origin of those cells. We haven't tried injecting just macrophages. I would speculate that pretty much any cell type that can produce DAMPs, that can produce cellular material when injected, is going to give us that immune response, but we haven't tested injecting in vivo one macrophage type or the other.
Can I ask something at this point? Did you, in your experiments, did you check whether the clotronate, liposomal clotronate, if similarly abolishes the resident and the circulating macrophages?
We did check that, and it actually it shows slight preference for circulating monocytes. It brings total cardiac tissue macrophages down about 50 to 60%, and this has been shown by some other groups as well, and that seems to be about the most efficient depletion that you can get with clotronate. But the interesting thing actually, and this has also been speculated, is that the macrophages that are not cleared, that are still full of liposome, actually also are functionally impaired. And so even if you're only depleting about 50 to 60% of the pool, you're probably having an even greater effect on their function beyond just that depletion. So we did look at that a little bit, not with sort of the more advanced techniques that you would need to do to really parse out tissue versus circulating, like you would need a genetic system to track that. But yeah, whole heart macrophage number was down about 50 to 60%. Emiliano Bidet, what about the NLRP3 inflammasome in this mechanism? Yeah, so that's another thing we're looking at. We haven't, that's what I'm in the process of digging into, really, is trying to understand how these various signals are working in macrophages, not just in cell therapy, but more in just how the macrophage really regulates and responds to tissue stress and tissue injury. So we're working on that. We haven't looked at it in cell therapy. Richard also asks, are the tissue resident macrophages already CX3CR1 positive? They are, and I'll point out two things. CX3CR1 doesn't just label macrophages. It does label a population of dendritic cells, a small population of NK cells, but it seems to be one of the major markers of the tissue macrophage pool. That said, it also does label a subset of monocytes, which are in mice LY6C low or patrolling monocytes. So CX3CR1 is a strong marker for tissue macrophages, but it is not a selective marker for just tissue macrophages. Anonymous attendee says, in one of your papers, you said that injection of a vehicle alone can induce immune response, which may be beneficial for heart healing. Could you please elaborate on that? Yeah, so we spent a lot of time comparing all of our data to saline injection because we were concerned that simply injecting the mice with a needle would produce enough inflammation to improve function. So we actually did a whole study where we compared saline injection to no injection, and we saw that the actual physical injection of saline and the inflammation associated with saline was not enough to improve function. So there does seem to be somewhat of a threshold effect. So the saline injection does give a small immune response, as you would expect, but it doesn't seem to be, certainly not to the magnitude of injecting cells or zymosin, and does not seem to be functionally enough to improve heart or to lessen heart repair. So we have that in our supplement for from the 2019 paper.
Ron, just another question or a comment here. You may remember, but probably you're too young, that something like 20 years ago, the surgeons started to laser to produce laser holes in the heart. Is that potentially a mechanism?
Yeah.
I mean, could what you have discovered now be the mechanism for this slightly obscure treatment? Because there were some benefits as well.
Yeah, I mean, it certainly seems to fit. And we've talked about other kind of ways that you could perhaps non-invasively induce this sort of transient damage. It's difficult to get people to buy into that now, I think. But yeah, I mean, it certainly fits with the idea that there is a certain sweet spot of causing some damage that could be beneficial. And the other thing that we always try to understand, well, why does the inflammation you see with heart injury by itself not lead to this sort of protection? And there's a lot of reasons for that. One of them might be that what we're seeing with this sort of reactivation of the wound healing cascade is without any additional damage, or at least significant myocardial loss. And so, you would think that you would need a mechanism that could produce that sort of transient damage, and has to be very controlled. I think we're probably a long ways away from being able to do something like that, but from a research perspective, it's certainly something I'm interested in.
But I guess, ablation to some extent does what you suggest. So for ablation for AF, and I don't think many people would argue that there's any regenerative potential following ablation.
Yeah. Let's see. Matthew Stratton says, have you tried this in aged mice? We have not. We're actually looking into sort of the question of timing, not in terms of age, but in terms of intervention. So what we did is we injected 7-day-old infarcts. And so one of the questions that often comes up is, well, is there a point of no return? So if you did that experiment and you injected a month-old infarct or a 3-month-old infarct, would there still be a benefit? So we're following up that. In terms of age, yeah, the interesting thing about that is that it's been shown that over time in mice, there's a shift in macrophage content, meaning that as the mice age, more of the heart's endogenous macrophage pool is replaced by monocytes. And so the speculation is that over time, that immune response not only might be different, but actually might be more pro-inflammatory. So it's an interesting question. We haven't tried it yet. Jake Kumar Pal asks, besides apoptotic markers, why pure apoptotic markers have not been evaluated in your study? Yeah, kind of goes back to the earlier question. We didn't see any overt suggestion of attenuation of myocyte loss. And so we haven't really dug into all the different modes of cell death that might be occurring. It's something that's certainly on the to-do list, but we didn't see any direct evidence that we were changing a large amount of cell death, in part because of the fact that we intervened a little bit after the primary infarct was formed. Let's see, Anonymous, did you address cardiomyocyte polarization differences between Zymosin-treated and/or MNC injected? I'm not quite sure what you mean by cardiomyocyte polarization. I don't know if maybe you were talking about proliferation, in which case we looked at that and we didn't see any major differences. We have not looked at
which actually, sorry, because we had this discussion, or just what Davor said. It's not the cardiac regeneration you're looking at. It's improved wound healing, right?
Right.
You have no evidence for any proliferation or regeneration.
Exactly. And I didn't show the data, but if you look,
that's important to state.
When we looked at the entire scar, we didn't see any change in scar size. So scar burden in these mice was the same. We just think that perhaps the ECM content of those scars is different.
But maybe this question was also addressing electrical properties of the myocardium.
Perhaps, yes. In which case we didn't look at that. Jeff Saucerman, hi Jeff. You mainly focused on CCR2 and CX3, CR1 positive macrophages as distinct populations. To what extent have you characterized the potential for transdifferentiation from CCR to CX3CR, any double labeling? Yeah, there is an overlap for sure. And we actually saw a shift from a largely CX3CR1 only population at baseline to a double CCR2, CX3CR1 double positive population with injury. And that suggests, based on the labs that have really dug into this, that those are derived from circulating monocytes. And so circulating monocytes can express both CCR2 and CX3CR1. Without a lineage tracing system, we couldn't really look at transdifferentiation. But a lot of the data that's out there would suggest that those are actually two discrete pools of macrophages, at least developmentally. And that the circulating influx of macrophages, they're going to be double positive. Absolutely. Jason Yang. Hi, Jason. Can you please comment on the heart's native wound healing capacity? And if you think this native capacity may set a threshold for MI pathology. Similarly, if you have an intuition, if the relevant macrophages are tissue resident or circulating. Yeah, so that's a great question. And we were talking about this a little bit earlier. There is speculation that as the heart ages, for example, the macrophage content without an inflammatory insult, an overt inflammatory insult, shifts. And the speculation is that that shift leads to a more pro-inflammatory and perhaps less efficient healing pool of macrophages. So I think that certainly could be playing a role. And as far as whether the relevant macrophages are tissue resident or circulating, so I think you certainly need both. And that's been shown by many groups. The tissue resident macrophages are really, to me, very interesting because they, for one, they're the ones that are there essentially throughout normal heart development and normal heart function. And the data that's out there does suggest that perhaps they have improved or a more efficient wound healing response. But no matter what, at the end of the day, they're not effective enough to impart a healing effect to any meaningful level with a large MI. And so, yeah, one of the questions I'm really interested in is why is that? You know, why if the heart has its own macrophage pool, why do you need this sort of emergency influx of monocytes? And if it was possible to improve the function of those tissue macrophages. So yeah, those are all questions that I'm very interested in. Rhea Stewart says, is there a difference in the response of inflammation in the fetal heart? Sorry? The fetal heart versus the adult heart failure, MI, yes. So as we were mentioning, the early embryonic development in fetal heart seems to have more tissue resident macrophages. And this has also been speculated by a couple groups, including Eric Olson's, that is a contributor to the neonatal repair capacity and regeneration capacity in mice anyway. David Wolfson, what was the reasoning behind investigating the CX3CR1 and CCR2 positive macrophages? Were there preliminary data that hinted these populations play a significant role? It really came from the literature on cardiac wound healing, and that as diverse as macrophages and other immune cell types are, we felt that those receptor systems gave us an initial inroad to sort of dissect major populations. And so certainly even within CCR2 positive macrophages, for example, there's lots of heterogeneity. Same thing with CX3CR1. So there's a lot more to kind of dig into, but we picked those because there were strong data in the field in the heart that those were major distinguishing receptors that we could impair and study. But sure, lots more to look at. Let's see, Adrian Arietta. Hi, Adrian. Concerning why the scar tissue has improved biomechanical properties, do you have any evidence suggesting that the collagen being deposited in the scar is undergoing some type of quality control that doesn't occur if the injected macrophages are not present? We don't. We're looking for that evidence. We're really interested in not just the amount of scar, but the actual composition, the integrity, the maturation of the ECM. So that's something we're actively working on. We did not see any major effects on alignment of collagen, but I think we have to really dig into that a little more.
Well, what about the type of collagen, by the way, Ron? Have you quantified that?
Not beyond what I showed, at least in terms of all the major collagens we've looked at were decreased. We haven't dug into that, and so what we're hoping to do is dig into that proteomically. So that's ongoing. Let's see. I was wondering. Manolis Salamondis. I was wondering if you assess the effect of the different macrophage responses that you observed to the vascular response and its effect to healing. The lymphatic response may be working in tandem with inflammatory cells to promote wound healing and scar resolution. Absolutely, I agree. We didn't, so we looked actually, and I didn't get a chance to show this. We looked for vasculogenesis from using our c-Kit cell reporter system, and we did not see major changes in vascularity other than actually with Zymacin, where we did see a significant increase in endothelial cells, but it was still not really large enough of a percent, we think, to improve vascular, to improve circulation. So you would see scattered, sort of pockets of single new vessels here and there, but nothing that suggests really, revascularization. Certainly, the immune response and the lymphatics and the vascular response are tightly intertwined. Especially CX3CR1, the interesting thing there, the ligand for that receptor is expressed pretty much exclusively in endothelial cells. So there's definitely some crosstalk there. We didn't see any major improvement in vascularity with our therapies. Anonymous attendee, what about Tregs? Did you see lymphocyte populations? Not really at the time points we looked at. I think a lot of what we were doing in the first 1 to 3 days might have been too early for that. We haven't dug into that long term, and that's something that again we're interested in. But we didn't look at Tregs to really be able to say. Same thing with neutrophils. We looked early and we didn't really see much of a difference in neutrophils, but likely there probably is something going on there. We just haven't found exactly the time point yet. Anonymous, Zymicin confers similar, if not more long-lasting cardioprotective effects than cell therapy. This begs the question as to whether we should really be investing so much effort into cell therapy versus small molecules like Zymicin. I like the idea of investing in immunomodulation as a therapy. Cell therapy has benefits in animal models, and I think what we've shown is that you can uncover some useful information. I think people have to try everything, but yeah, I think cell therapy is, I think we could certainly be doing a more mechanistically driven approach.
Did you have a chance to look because the next horizon is exosomes or micro vesicles, whatever, and it's tempting to speculate with it, maybe similar mechanisms applied to this kind of therapy. Did you have a chance to look at that?
Not direct. We didn't directly look at exosomes or microRNAs. So it's possible that you can actually change the wound healing response intrinsically, and that you might actually change, what the cellular milieu is in that wound. We think that the fact that you can induce this inflammation without a cell and get this benefit would suggest that it doesn't have to come from the donor cell type. Doesn't mean that couldn't be a mechanism, but it seems like that immune response is the ignition stimulus. As to what occurs downstream, we think fibroblasts and fibrosis is an important part. Certainly could be other things, but yeah, we didn't look directly at exosomes as an immuno activating stimulus. Nicole Pianca, if I am not wrong, the beneficial effects of cell therapy is almost due to remodeling of the ECM and effects on non-cardiomyocyte cells. You said that there is no increase in cardiomyocyte proliferation. Yes, from what we've looked at, no increase. Do you find any other effects on cardiomyocytes? Is there an increase in functionality? Do you think that there could be a crosstalk among ECM and cardiomyocyte? Yeah, so great question. And what we didn't really look at was the surviving myocytes in these infarcts. And so if you change the ECM, for example, you could very easily envision that changing, the stiffness properties of that tissue, and therefore changing the mechanosensing of the cardiomyocyte. So I think that's absolutely potentially in play. We didn't look at that yet. But yeah, and then the other thing is that these were reperfused infarcts, so the tissue was a heterogeneous mix of scar and myocytes, which would suggest again that crosstalk could be a major player for sure. Yeah. Svetlana Riley, how widespread is the acute inflammation in your experience? Is it strictly localized to the area of injection, or does it spread throughout the whole LV-RV? Great question. Would your data suggest that the localized stimulation is sufficient to improve function? Yes. So when we injected cells or Zymicin into healthy hearts, we were able to localize that within, I want to say, 20 to 30 microns of the area of injection. And so you could see these really distinct foci where these injections were done. And if you actually looked across the whole tissue globally, there was not a significant increase in immune cells. You had to look in that area of injection. And actually, by 2 weeks, that injection area, the inflammation will resolve and it would leave a scar. It would actually leave a scar you could find. So the cell therapy effect absolutely is localized. In terms of would localized stimulation improve function? So our data with the infusion model would suggest that, yes, you do have to induce inflammation somewhere around the area of the wound or the border zone. The interesting thing is that in rodents, the area of infarct is, we think, small enough that 1 or 2 injections is sufficient. You start scaling up to large animals, the data that's being put forth would suggest that you need multiple injections. You need 10 to 20 injections at minimum across the whole border zone. So there certainly is a physical distancing effect that probably is playing a role here. But in the mouse heart and the rat heart, it seems like those sort of border zone spanning injections are sufficient. Richard Heads, so if DAMPs are involved, does that mean TLR4 is pro-reparative in tissue-resident macrophages as opposed to pro-inflammatory or neutrophils? I can't get this idea funded. I can't help you with that. I think, so we're very interested in toll-like receptor signaling, and we think that, so zymosin actually induces TLR2, I believe, primarily. But toll-like receptor signaling and modulation of that in the right context, either in different cell types or in different amounts, certainly could be beneficial or detrimental. And so this is something that, again, we're starting to dig into. We haven't done anything directly to look at TLR4, but it's a major signal in the heart for sure. And so I would agree that there is probably a window of benefit to stimulating TLR4.
You would give the money.
I don't get to make those choices, but I would be intrigued. Let's see. I think this is the last one. Rio Juni. Prolonged inflammation is also detrimental to the heart. Do you think there's a time window when you would want to activate the immune response and then deactivate it before it induces damage? Absolutely, and obviously that's the million-dollar question. Yes, that would be the ideal. How to do it, how to achieve that, it's going to be challenging. Too much inflammation is almost universally detrimental. And so I guess kind of going back to the previous question, it is a little bit of a counterintuitive concept, causing damage to improve the damage response. And so we think that certainly you have to be able to control this if it's going to ever be therapeutic. I think we have a lot more research to do. But from a basic mechanistic perspective, there's a lot to learn here. So
there's yet another one, a personal one.
Great talk. How does it feel to publish in Nature? It's obviously I'm very thankful, but it is it's a little stressful too.
I'm sure there are worse things in life, right?
Certainly worse things in life,
I guess.
Well, thank you all so much. I hope I was able to provide some food for thought.
I think you did a great job, both in your presentation and in answering the questions. I learned a lot.
Thank you. Thank you, everyone, for tuning in.
Okay, great. So tomorrow, Peter Ferdinandi.
Yes, and I would just like to once again, just share my screen and just thank the ISHR and actually just remind the younger audience in particular about the JMCC Junior Reviewer Program. Apply, great experience. You'll probably learn a lot, I'm sure, and you'll meet some wonderful people as well. So I'll stop sharing now. And I would like to thank both of you once again. Fantastic webinar, Ron. Fantastic chairing, Thomas. And it was a real pleasure.
Thank you so much.
Thank you.
And thank you to everyone who tuned in. See you all tomorrow.